Hello. So, hello and welcome everybody. Have a seat if you can find one. Otherwise, stand up. And I hope you got filled up with snacks so you can get it through this really exciting lecture session. So my name is Carola Hesemann, and I'm studying biotechnology here at DTU on my master's. And I'm in this student organization, SBE, Society for Biological Engineering, which is hosting this event for you today. And you can see the program here, which you probably already did. So this lecture session is sponsored by two sponsors. One of them is IDA Young Professionals, and then we have Danish Biotechnological Society, so you can thank them for all the snacks and soda that you just got. And as I said, this lecture session is hosted by SBE DTU, and you probably some of you saw the flyers on the chairs. We are a student organization from students to students, and we have different events. For example, these lecture sessions, and also Reach and Discover, which is a big event coming up. It's the 12th of November, and I encourage you all to go there. It's really, really nice. It's a project conference for students, so if you don't know where to write your bachelor or master thesis, you should definitely go there. You can meet all the researchers here at DTU and some from industry as well. Then we have company visits to different places. We have Christian Hansen in next week, and then we have a mentor program as well and other events. And if you want to help us out doing these, hosting these events, you can always come to our member meetings or find us on Facebook. And you can see the next member meeting is the 5th of November. You can see it on the flyers lying around, or you can always talk to us with the name tags. So, yes, now I just want to give the word to Christian Honoré from Novo Nordisk. So, yes, give him a hand. And
Okay. Sorry. It's really loud. Okay. Well, thank you to Carola and the organizers for inviting me. And thanks for the big turnout. I'm very excited to see how many people are interested in stem cells. But before I get started, I was just encouraged to say a few words about my background. So I have a degree in biochemistry from the University of Copenhagen. I graduated back in 2005, and I did my master thesis at the University Hospital of Copenhagen, working in the innate immune system, something completely different from what I'm going to talk about today. But after my master thesis, I was fortunate enough to continue my studies in the same lab, doing a PhD, and so I continued with the same research, which was great, and I learned a lot. But coming to the end of my studies, I also realized that I wanted to try a new research area, and also I really wanted to go abroad. So I started looking for opportunities, and I was very fortunate to get in touch with a professor in Boston who was working with stem cells and directing them to beta cells. And even more fortunately, I got a position in his lab, and I went there for a little over 2 years, and I learned a great deal about stem cells. And then I came back in end of 2012, and I got a position at Novo, which is the position I'm in today. And really what I'm doing at Novo is trying to evaluate the use of induced pluripotent stem cells for modeling various forms of diabetes, and sort of evaluating this technology to see whether we can use that for our drug discovery and development efforts. And that's what I'm going to be highlighting today. So there are multiple forms of diabetes, but it all comes down to the patients not being able to regulate their glucose levels. And the glucose is regulated by the beta cells, which is the cell illustrated here. And it does so by sensing the levels of glucose in the blood. And once that glucose gets above a certain threshold, the beta cells respond by, can you see the pointer at all? No. Well, the beta cells respond by secreting insulin. Insulin is the hormone, as you probably know, that regulates the glucose. It goes out into the periphery, tells all the tissues to take up glucose from the blood. And that obviously results in a lowering of the blood glucose. And once you get below the threshold, the beta cells will stop secreting insulin. So that's how it regulates, very simplified, the glucose levels in the blood. In type 1 diabetes, one of the forms of diabetes, the immune system actually decides to attack the beta cells for a reason not quite known. So you lose beta cells, and hence you lose the capacity to regulate your glucose. In type 2 diabetes, you often have defects in the beta cells, but you also have peripheral resistance to insulin. So the glucose levels goes up, and that puts a higher demand on the beta cells. Ultimately, these beta cells become stressed, and they die. So in type 2 diabetes, you also end up missing beta cells. And then you have a third group of diabetes called, it's a big panel of diabetes. It's called monogenic forms of diabetes, and what that is really covering is it's just multiple mutations or single mutations in single genes that plays a key role in beta cells. So there you have a dysfunctional beta cells. But as you can tell, it all homes in on the beta cells. So if you want to learn more about diabetes, you want to study the beta cells. Unfortunately, beta cells are not that accessible from human beings. They're located in small clusters of cells called islet of Langerhans within the pancreas, and you can't just go in and take those out from a human being. So we're limited to beta cells from cadaveric donors, and we can't really control whether these donors have diabetes, don't have diabetes, or what their genetic background is. So we've been sort of limited to just using animal models and the beta cells we can get from cadaveric donors. So there's a great need for other human models for studying beta cells. And that brings me to these induced pluripotent stem cells, or iPS cells, as we like to call them. So in 2006, Shinya Yamanaka demonstrated that you could take a human, or a mouse in this case, and then later on a human skin biopsy, a somatic cell from a human. You could overexpress 4 transcription factors in those cells, and then revert them back into a state that's very similar to a pluripotent stem cell, which is the state here. Pluripotent stem cells are quite unique. They have 2 very interesting capacities. They can proliferate, and they can proliferate, at least in theory, unlimited, so you can get a huge amount of cells. And they also hold the capacity to differentiate into all cell types of the human body. So you can imagine that you can take a skin biopsy from the patient of interest. Let's say it's a diabetic patient. You can reprogram that back into an iPS cell. You can then take that iPS cell and differentiate it into the cell of interest, which is in our case going to be focusing on the beta cell. And then you can actually start looking at these beta cells that you're deriving from the patient-specific iPS cells and compare them to beta cells that you're deriving from healthy controls. And that's really what we mean by disease modeling. We're comparing cells derived from iPS cells from patients and controls, and to see if there's any differences, and can then help us learn more about this disorder. And if we find phenotypes that can educate us on the disease, but also use those phenotypes for drug screenings, where we're using the phenotypes to identify compounds and drugs that can alleviate this disease phenotype. And ultimately, or at least the ideal scenario will be then we can use that for developing better drugs that can go back into the patient from whom you derive the iPS cells from. So that's sort of the broader picture of iPS cells and for the in vivo or in vitro disease modeling. What I'm going to be focusing on today is the first 3 parts of this diagram. I'm going to be talking a little bit about the derivation of iPS cells. I'm going to be spending a lot of time talking about the directed differentiation of these cells towards the beta cells, and then also finally finish off with a little bit of the disease modeling part of this. So we're very fortunate to be part of a big collaboration that spans across Europe, where we get access to patients, different diabetic types of patients, the different diabetics we're interested in. That's both monogenic forms of diabetes and also type 2 diabetes. The clinicians take the skin biopsies from the patients, send them to another collaborator who derives the iPS cells, again by overexpressing these 4 transcription factors. And then we get the iPS cells, and we do a bunch of characterization on these cells. And that's mainly just to ensure that these iPS cells really are pluripotent. And one of the things we do is we look for markers that we know should be present in these iPS cells, and that's the panel to the left, where we are staining for multiple markers that we know should be present in a pluripotent stem cells, and in most cases they are. We also ensure that these cells can differentiate into all 3 germ layers and also derivatives of, and that's shown up here in the top panel. And then we ensure that we have highly pure cultures and also that they're karyotypic normal. And in most cases, these iPS cells are of really good quality, both whether you derive them from a patient or from a human or from a control individual. So we can get iPS cells from the patient. The next step, obviously, is then to differentiate these cells towards the pancreatic lineage. We want to generate beta cells from these iPS cells. And conceptually, directed differentiation is pretty simple. You have your pluripotent stem cells, and you want to get to the cell of interest, which in our case is the beta cell. It's pretty simple. You're going from A, and you want to get to B. But then you look at all the pathways that these cells can decide to take, and then you realize, okay, it's not very simple. And I can tell you from working 5+ years with these cells, it's not easy, but it's also extremely fascinating. So don't be discouraged by this diagram here. And it also shows you can really select your cell of interest and try to get at it. But obviously, we're interested in the beta cells. And we've also been very fortunate that we've learned a great deal over the past decades from developmental biology. We've learned how beta cells develop in animal models, primarily in the mouse. And that's the top panel illustrated here. That really shows how a beta cell develops in a mouse and also most likely in a human. And that's really allowed us to define distinct stages that we know our cells have to go through. And what's even more cool is that we can use some of the markers and some of the genes that are expressed at these distinct stages to really sort of classify or characterize our differentiations and how good are we at differentiating the cells. And even more neat is that some of the pathways and genes that are involved in a mouse towards the differentiation of the beta cell also translates to the human system. So we can take some of the signaling molecules that works in a mouse and actually put them on a human system, which is really cool. And you can see that in the bottom panel. I've tried to just illustrate the distinct stages that we want our cells to go through to get towards what resembles like a beta cell. And as you can see, the first thing we really want to do is take our undifferentiated cells and then generate definitive endoderm, which is one of the 3 germ layers. And we know definitive endoderm gives rise to your whole gastrointestinal tract. So that's the first step to get to that. And shown here is a culture of undifferentiated iPS cells where I've stained for a marker called OCT4. That's a transcription factor we know is present in pluripotent stem cells. I've also stained for SOX17, which is present in definitive endoderm, and you can see there's nothing there. And the blue marker is just labeling all cells. So that's the starting point. And then after 4 days, what we're getting is something like this. You can now see that the picture is completely reversed. We now have cells expressing SOX17 and also other markers of definitive endoderm, and hardly any OCT4 positive cells. And when quantifying that by flow cytometry, you can see we're again looking at the same markers. You have OCT4 on the x-axis and SOX17 on the y-axis. You can see that iPS cells, which is to the left, all of them are OCT4 positive and none are SOX17 positive. And then again, after 4 days of differentiation, we have above 90% of the cells expressing SOX17. But as I mentioned, definitive endoderm can give rise to your gastrointestinal tract, your liver, your lung, your pancreas, your intestines. So we want to specify the region from where the pancreas develops. And that's really illustrated by the next panel here, where we're trying to specify it towards the pancreatic lineage. And here we're using 2 transcription factors, PDX1 and NK6.1, that we know are crucial to developing the pancreas. And again, quantifying by flow cytometry, you can see we get a very nice induction of PDX1. The left panel is the definitive endoderm. There's no PDX1 expression. And then after 4 to 6 days of differentiation, we now have above 90% expressing PDX1. And also, the induction of NK6.1 comes on a little bit later and not at the same efficiency, but still pretty good. The reason why I'm highlighting this population is because it's been very interesting, and for the past several years, a lot of groups have shown that you can take this cell population that we call pancreatic endoderm, you can transplant it into a mouse, a diabetic mouse, and let it sit in the mouse for 3 months approximately, and they will actually differentiate and develop into functional beta cells that can rescue those mice from diabetes. So we know within this population of cells, we have the competence to generate beta cells, but these are not beta cells just yet. So we want to do that in vitro, in a dish, not put them into a mouse and have them mature. So we were very fortunate that this past year, 2 publications came out that showed how to differentiate these cells further towards something that resembles a beta cell. And shown here is our cells that we've differentiated. Well, let me start with this one here. These are human islets, the small clusters of cells that contains the beta cells from a human being. And what we're staining for here is C-peptide on the x-axis. C-peptide is a co-product of insulin. So if you have C-peptide, you also have insulin. So these are your beta cells. Then you have glucagon on the y-axis. That's a hormone produced by alpha cell, another cell type present in these islets. So you can see in this specific prep of islets, we have approximately 33% of the cells being beta cells. And then you look at these cells I've labeled stage 7 cells. Those are IPS cells. They start off IPS cells. 1 month later, these are stage 7 cells. So that also tells you a little bit about how long this takes. But you can see we have approximately the same number of these insulin-producing cells at this stage. So we're getting closer to something that looks like a beta cell. And that's also illustrated by some of these immunofluorescent images I've shown here. You can see on the left panel, we've stained for glucagon, again, this hormone produced by alpha cell, and C-peptide, which marks insulin production. And you can see we have cells both expressing glucagon and C-peptide, probably representing more immature cells. And then we have cells only producing one or the other hormone. So we're generating endocrine cells of the pancreas. And another key point is, again, this transcription factor called NK6.1. We're very focused on this one because we know it needs to be there to have a functional beta cell. And again, we get cells expressing that along with insulin. So that's really exciting. And we're getting much closer towards a beta cell. We don't have a fully functional beta cell in the dish yet, but we're definitely getting closer. So that's very exciting. And that brings me to the last part of this talk, which is the disease modeling part. So now that we can actually take our IPS cells and differentiate them very close to a beta cell, we obviously want to start looking at this disease modeling. And one example I'm going to highlight today is a rare syndrome called Wolfram syndrome, which is caused by mutations in the wolframin gene. It's quite a nasty disorder, and the patients who have this develop diabetes at around 6 years of age. And this is thought to occur due to death of beta cells. So we thought this would be a good model for us to study. Wolframin encodes the protein shown here, which is a large protein that is located in the endoplasmatic reticulum of all cell types in your human body. But it's highly expressed in beta cells. So we know it needs to be present in beta cells. And it's also thought to regulate ER stress, which makes sense because in the beta cells, you produce a ton of insulin, and that goes through the ER, stresses the ER. So you really need to have some mechanism that regulates the ER stress in these beta cells. And wolframin is thought to do so. So to go further into this, we actually identified 2 patients, 2 siblings, that had mutations in this wolframin gene. It was a compound heterozygous, meaning that there was one mutation on one allele and another mutation on the other allele. And both of these mutations led to a pre-termination stop codon, so they had truncated forms. And we derived IPS cells from these patients. And then, as I mentioned, wolframin is expressed in all cell types, but highly expressed in beta cells. So we just started off by looking at the undifferentiated cells to see whether we could detect this protein in these cells. And shown here in the diagram here is just looking at the messenger levels of wolframin. And you can see that compared to the housekeeping gene, we have levels of wolframin mRNA that's quite similar between 3 control IPS lines derived from 3 healthy individuals and the 2 IPS lines derived from the 2 wolframin patients. But then you look at the protein levels, you can see it's a different picture. And again, in the 3 control IPS lines, you have the full-length wolframin expressed. But in the 2 patient-specific lines, we have no protein. So basically what we have here are IPS lines that have a knockout for this gene. So as I mentioned, wolframin is thought to regulate ER stress. And I won't go too much into details about the mechanisms behind ER stress, but this shown here is the unfolded protein response. And really what that does, it senses accumulation of unfolded proteins in your ER. And when that happens, these 3 pathways called ATF6, PERK, and IRA1 can sense the accumulation of unfolded proteins. And that will lead to a downstream signaling cascade that ultimately just shuts down transcription to just stop protein production in that cell. But there's also a specific translation of certain chaperones that will go back into the ER and try to alleviate this accumulation of unfolded proteins. But there's a triggering point here that if the unfolded proteins sustain or are present for a long time, the UPR will shift and then we'll actually start killing the cells just to say, okay, this is going wrong, we're just going to terminate. And that's really what's assumed to happen in beta cells. And more specifically, wolframin is thought to regulate ATF6 levels negatively. So really with that in mind, we started looking at the ATF6 levels in our iPS cells. And shown here are a Western blot looking at ATF6 levels in, to the left, the patient-specific lines, and to the right, the control iPS lines. And I've loaded two different protein concentrations, but if you just focus on the lower concentration, the two red arrows here shows the ATF6 levels in the patient-specific lines. And here you're seeing the ATF6 levels in the control lines. And it looks like, at least this is a tendency, we haven't confirmed it completely yet, but it looks like we have more ATF6 in the patient-specific lines, which correlates well with what we're expecting, that wolframin regulates ATF6 negatively. We've also tried to stress these cells with chemicals to see whether we can induce the stress response, and what we're seeing is we're getting a higher stress response in the patient-specific iPS lines compared to the control lines. So that's obviously on the undifferentiated state. The next things we want to do, we haven't really gotten to that yet, is really differentiate them towards the beta cells. And what we can see is we just take a control line differentiating it towards the beta cells. We start seeing both upregulation of the mRNA and protein for wolframin. So that's, again, along the lines where we're expecting that we have a high expression of wolframin in the beta cells. And the next step, obviously, is then to do the same experiments in these cells. So that's really what I wanted just to show you today, and sort of what I'm hoping my point has been is what we can use these cells for. We're just getting started, so I don't have the perfect example. I can give you an idea about what it is we want to do with them. But as I mentioned, we can generate iPS cells from patients and also from healthy individuals, and we can really identify the patients of interest that we want to study, and then we can differentiate these cells towards something that resembles a beta cell. It's not quite a beta cell yet, I want to stress that point, but we're really focusing on figuring out how similar are these cells we're generating in a dish compared to a human beta cell. And finally, I just showed you some preliminary data on this disease modeling aspect of the beta cell, or of these iPS cells, where you can generate cells of interest and compare them patient versus controls. And ultimately, we, of course, want to use that for learning more about the various forms of diabetes that we want to treat, but also can we use this to test our drugs on these cells to see whether the phenotype we're looking at in the dish, can we alleviate that phenotype? So I'll stop with that and just thank some of the people who carry out the work and also some of my collaborators. And I'll take any question. Thank you.
Do anyone have any questions to Christian? Oops. Yes?
I was wondering how important it is to have more present iris along the alpha cells and not the beta cells in order to study these cells. Is it really enough just to look at the beta cells?
No, I can just repeat the questions for the people, but it's whether you want to build a whole islet composed of the different endocrine cells, or you just want to focus on the beta cells. I guess it comes down to the question you want to ask, because some of these forms of diabetes also affect the other endocrine cells, and these cells definitely crosstalk. So ideally, you want to build a microorgan in the dish if you can do that, or you can also look at it the other way. You want to direct the differentiation towards the individual cells and then try to aggregate that afterwards. We're not that good at it yet, so we can't really control whether we're generating one or the other cell type.
Any more questions?
I guess I was very clear.
Yes. But otherwise you will stay afterwards and you can go and talk to him. So yeah, we have a little present for you with the
Thank you.
penicillin microbe and some goodies.
Thank you.
Yeah. So thank you very much, and give a big hand to Christian. So our next speaker is Carla Christine Freude from Copenhagen University. Yes.
So the pointer's not working right.
No, the laser can't reflect on it.
Okay. Can everybody hear me? I guess yes. It sounds very loud to me, at least. I'm here and want to talk about induced pluripotent stem cells for disease modeling, and my focus is on neurodegenerative diseases. But the principle is very similar, like what you just heard. So a lot of it will be, not a lot of it, but there will be some repetition also from the previous speaker. And first, I was supposed to give you an overview of my background. So I'm originally from Germany, and I started doing a master degree at the Robert Koch Institute in Berlin, Germany. And I was working with mycobacteria, and we wanted to see mycobacteria, you know, there's this one famous strain which causes tuberculosis, and we compared it to a non-virulent strain and wanted to see what the difference in gene expression is there. Then I got bored with bacteria because I wanted to do something which is more related to humans. So I moved on to the Max Planck Institute for Molecular Genetics in Berlin, and I worked on X-linked mental retardation. And that was breakpoint cloning, translocation, so also very genetics and very limited in modeling diseases, because it was only a few lymphoblastoid cell lines we were working with, so it was not much of modeling. And then I moved on to America. I went to California, and I was working on diabetes there, actually type 1 diabetes. And I focused on 2 genes. One of them you heard was NKX6.1, which is important for early beta cells. And also I was working on another gene which was not mentioned, but is very important to make the pancreas, and that was SOX9. And you can see here a mouse pancreas, you can't point. And on the left side you have the wild type, and if you knock out the gene, you have hardly any pancreas anymore. So that was more disease modeling and mouse models. And then I did a second postdoc because I was always very interested in neurodegenerative diseases, and I worked on Alzheimer's and Huntington's disease. And then finally I came here, and now I'm working at the University of Copenhagen. I'm working on Alzheimer's disease, frontotemporal dementia, and model these diseases in the dishes with using iPS cell lines and differentiating them into neurons. So it was a long trip, but now I'm here. So maybe later, if you want to have some input on how to choose a lab or what to do, or how to streamline your efforts, you can also talk to me. I might give you some interesting stories about that. Okay. So we're interested in neurodegenerative diseases, and we're doing basically the same thing like a lot of people are doing. We're collecting fibroblasts, so skin cells, from the patients. We're making iPS cell lines, and then we're differentiating them into neurons. And then you can do different things with it. You can correct the mutation by gene editing. I will go later back to that. That could potentially be a cure. But as you might envision, if somebody has Alzheimer's and you're correcting the mutation, you put it back into the brain, it's not really a workable solution right now. So we're very far from this part yet. But what we can do right now is we can look and investigate the cellular and molecular disease mechanisms. And we can also do drug screenings. We can also do customized medical treatment, which is obviously sounds very good, but it's also a question of price, so it's always good to work on a disease which is very common, like Alzheimer's. This disease is very interesting, but not very common, and this will be my example for today. And it's frontotemporal dementia linked to chromosome 3, or short FTD3. And it originated in one very unfortunate individual, which you can see in this circular pedigree. So one woman acquired this mutation in CHIM2B, which is the gene affected in FTD3, and passed it on to various children she had, and they passed it on further. So then you end up with a lot of individuals which have this mutation originated just from one person. And this family lives in northern Jutland, so it is a Danish disease, if you might want to call it like that. But there is another disease, another mutation has been found in Belgium. It's a small family, so there's just this large family in Jutland and a small family in Belgium. So that's very interesting for research, but nothing which you can really use in industry. But what are the symptoms of these patients? So here you see a brain of a patient with frontotemporal dementia type 3. And the title already gives it away. So what you see is that the frontal lobes, but also the parietal lobes, are disappearing because the neurons get degraded, they die off. So you have global cortical and central atrophy. They don't have these features like AD patients. They don't have plaques or anything like that. They have behavioral changes, which is also very various between different individuals who are affected. So they can be very apathetic, or they can be very restless. They also have something which is called disinhibition, so they cannot control it. So like if they're angry with you, they would basically just call you names. They all have hyperorality, which means they like to talk a lot, and they don't stop. Later, though, they turn into mutism, which means they don't talk anymore, and they also have dystonia. So what is wrong with these patients? You can see here the protein which is affected, which is called CHIM2B, charged multivesicular body protein 2B. And in the wild type situation, you have a normal protein with 213 amino acids, but then the mutation is at a splice site. So you get two different transcripts made in the patient. So you have one which is called intron 5, which is basically short 177 amino acid plus one extra amino acid, which is not found in the wild type protein. Or you have another one which is called delta 10, which is again the same 177 like in the normal patient, but an extra novel 29 amino acids. And then you have various contributions of these truncated proteins. So these proteins, the mutation doesn't cause that the protein is absent. The mutation causes a novel abnormal protein, so the C-terminus of the protein is changed. And thereby it classifies as a gain-of-function mutation. So what does it do? CHIM2B is part of the endosomal sorting complex, and this complex has two major functions. So first of all, these endosomes are fusing with a lysosome, which is there for degradation of proteins, but they're also involved in recycling receptors on the cell surface. So that's also something which is very important for nerve cells, because they communicate via receptors. And CHIM2B is part of this complex. So when you have a mutant CHIM2B, the fusion of the late endosome and the lysosome is not working anymore. So you get a really enlarged endolysosome, and this whole pathway is blocked. So you might wonder why patients are still alive, because this frontotemporal dementia happens at the age of 50-ish, 60-ish. But the reason is because they also still have a normal copy. So it's all about sensitivity of the cells to this faulty mechanism, and also still having a normal copy which can cope for a while with the problem. So here you see brain sections, and what is encircled here, you have two patients, and you see these bubbly structures in the brain compared to a normal control on the side. So they have these enlarged endolysosomes which you can actually visualize in the brain. And as you can think, there is a special role of endosomal trafficking in neurons, because the architecture of a neuron is very complex. So they have complex dendritic arbors and extensive axons. So there are special challenges for intracellular signal transduction, because it has to go from one side to the other. So this endosomal system enables this trafficking of these signal molecules through the axons and over really long distances also, because some axons are really long and extensive. So the endosome system is fundamentally equal in all cell types, as I said, because you have this mechanism in every cell. But obviously the neurons are especially vulnerable to problems in this system. So what happens if you have this problem with the malfunctioning endolysosomal system? You have a decreased cell survival. Also, your axonal outgrowth due to missignaling and guidance is not working, and the dendritic branching is also inhibited, so the dendrites cannot branch, and overall cell migration is inhibited because of malfunctioning signaling. So that is something very similar to the talk before. So what we did is we took the patient cells, because obviously we want to mimic the disease in a dish and maybe get some new insights into this pathway and into the disease. And we took the approach of taking iPS cell lines and used a non-integrative system to make iPS cells. So these cells are transduced with episomal plasmid. That means that these plasmids do not integrate into the genome. They just deliver the pluripotency genes, convert the cells from fibroblasts to iPS cells, but then after several passages, they get diluted out and they disappear. So you have non-integrative methods, which is preferably because you never know if a phenotype you see is caused by integration of your transgenes. So thereby you have no integration. And then again, you can see that we check that the mutation is there, karyotype is normal, so all the usual you need to do when you're making iPS cell lines. And here you can see, similar to the previous talk, you check for the pluripotency markers that your genes, that your cells have become iPS cells, so that they're truly pluripotent. And then you do something which we call in vitro differentiation. So we take the cells and we expose them to some maturation media, which is usually just fibroblast media, and see that they have the potential to develop into all three lineages, because the whole organism is basically made out of these three basic lineages, which is ectoderm, mesoderm, and endoderm. And when you can do that, you can say, yes, my cells are pluripotent, I can do everything I want with it. And for the ectoderm, you usually use beta-3 tubulin, which is a neuronal marker. Mesoderm is smooth muscle-actin. And for endoderm, you can use AFP, which is alpha-fetoprotein. Another thing which we started now, because when the field moves on, it's very difficult to use age match and gender controls, because you don't really know the background of these patients. So to have a really clean control, we go in and we specifically repair this single nucleotide mutation, because then we can directly compare the patient with an isogenic control and can see this is really due to the mutation and has nothing to do with any genetic or epigenetic backgrounds. And the method we're using is CRISPR-Cas9 mediated gene editing. And this is a very elegant and very simple method. So you have your target DNA, which you want to either mutate or repair. And then you design something which is called single-guided RNAs, which specifically bind to your region of interest. And they also need these so-called PAM sites, which are important for the protein to be directed to the location of action. And the protein which is important is Cas9. And Cas9 has been derived from bacteria strains, where it is actually some sort of immune system of bacteria, because they are destroying foreign DNA. So the single-guided RNAs are directing this Cas9 protein, which you also have to deliver in form of a plasmid, overexpressing plasmid, into your cells. And then this Cas9 makes a double-stranded break. And all you need to do is give a template, a short template of nucleotides, so that the cell can do a repair. So it either does then a homologous recombination, which would mean that it would repair the mutation or insert the mutation you want, Or if you want to make a knockout cell line, for example, you could use the non-homologous end joining pathway, so you don't provide a DNA template, and it would just cut it there, and it would be broken, and it would randomly repair, so your gene would not be functioning. But we use it to make isogenic controls, so repairing the mutation in these cases. So our tools for disease-specific studies are the patient-specific iPS cell lines, the isogenic controls, and also we're looking into age and gender match controls. But a lot of these diseases, since they're age-related, it's difficult to really know if it is an age defect of the cells or the patients, or if this is a true pathology. But then again, what you have to do, so now you have the iPS cell lines, but what we want is neurons. So we need to go down the path of differentiation from making iPS cell lines into neuronal progenitor cells and then differentiating them either into astrocytes or neurons. We do not want to have endoderm or mesoderm. That's not what we want. So our protocol is we're using these iPS cell lines, and then we're using inhibitors, which are basically can't really show it here, which is this A8301 endorsomorphin, and these inhibitors basically just block mesoderm and endoderm, so you're getting down neuronal differentiation. And then you're adding certain factors which are encouraging neuronal differentiation, and you end up with something which is called neural progenitor cells. And then with these neural progenitor cells, you have to further treat and mature them into these specific subtypes of neurons. And again, this is one of the biggest problems when you're studying cells, is that you don't get a pure population. You get maybe 80% of cholinergic neurons if you're lucky, or if you want to have glutamatergic neurons. But you will also always have other cells in there, so you never get completely pure populations. But you want to at least enrich for the neurons which you are interested to study. So this is just to come back to what the problem is. So in our patients, we have this mutation, which is basically keeping the late endosome and the lysosome fused together and extremely enlarged. So what we then do, or did, is we're looking at the patient, and these are iPS cell lines, so it's very early. It's not even neurons yet. And when you look at this, this is Cd63 staining, which basically stains these late endosomes and lysosome complexes. You can see these green, bigger bubbles in FTD3, whilst in the control you don't see that. And then we did some advanced confocal microimaging and counting and stuff, and that really shows you, okay, there is really an increase in these enlarged endosomes in the patient compared to the control. So we can see this disease phenotype I showed you early in the beginning in the patient brain also already in our iPS cells. So we find that very exciting. And then we went on to make neurons. And now comes the tricky part, because we're interested in forebrain neurons. So we want to get forebrain neurons which are preferentially glutamatergic. So what you do then, you do follow this differentiation protocol I showed you, and then you have to stain these neurons, or NPCs, for specific markers. So for nestin, FOXG1, OTX2, and PAX6, which basically show you you're going down the right way into forebrain neurons. And then when you mature them, you want to show that these cells are neuronal. So there's different markers for mature neurons, like MAP2 and TUJ1 and tau. But other markers, like for example GFAP, that marks the astrocyte. So you don't want to have too many of them. But then again, you need a minority of astrocytes in your culture. Otherwise, the neurons are not very happy. And then you stain for V-glut, which is basically indicating that you have glutamatergic neurons. And then you stain also for GABA, which is inhibitory neurons, and see how many you have these. And then you don't want to have dopaminergic neurons, because we're working with frontotemporal dementia. Dopaminergic neurons, which is dopamine would be TH, the marker. You don't want that. But if you're studying Parkinson's disease, for example, you have to do a different protocol, because you want these and not the other neurons. So it's very dependent on what disease you're looking at. And then during your protocol, you also want to see, am I in the right layer? So you're looking, the cortex is divided into different layers, into 6 different layers. And you want to see that ideally you would produce these neurons which are in the layer which are mostly affected in the patient. And with our protocol, we're getting mostly deep layers, like marked here by TBR1 and CTIP2. So not the superficial layers, which can be, which is most likely due by the amount of differentiation. Some people do differentiation for several months to get really mature neurons. And in order to have a functional disease model, you also need to know that your cells are really doing what they're supposed to do. So what you can do then is a very sophisticated method, which is single-cell patch clamping. So you're basically giving your neurons an electrical stimulation and see if they can produce action potentials. And if they can do that, you can claim that they are at least basically functioning. So we all know, or I just showed you before in the patients, and we could also see it in the cells, that there are cellular abnormalities in these patients. So we were interested in looking a little bit more detail with electron microscopy. And what you can see here is a diseased neuron, and it's basically this whole structure here, and you can see these actin filaments, which are a little bit disturbed. And in the end, down here, you can see these endosomes, which are really accumulating and disturbing the whole process. And if you, what did I do? If you compare it, or if you look a little bit closer, this is a different neuron, but you can see here also you have these large vacuolar structures. So that's also possible to really visualize that with electron microscopy. But what was really interesting and novel to us was that we found these bizarre mitochondria structures now. So these structures in the patient, they are not very well formed. So the cristae in the mitochondria of these patients are not really well defined. Because if you look at a patient, not a patient, a control, you can see that there's very nice cristae and very nice distribution of mitochondria. So not only did we find this enlarged endosomes, which is basically already has been shown in the patient itself, we also saw that there is a mitochondrial defect. And then we went on further to do some Cos experiments. And the person who performed this experience is actually sitting in the audience. And Cos is basically a machine which shows you what the normal level of oxygen consumption is in these cells, but also what is their potential. So what can they actually, what's the maximum oxygen consumption possible? And here we have an example where we look, the lower line is the patient. So you can see they already have a lower oxygen consumption, and the maximum capacity is also reduced. And this problem can be completely fixed by using the isogenic controls. So you're comparing the patient to its isogenic control. So that looks like this is really a disease-specific phenotype here we're seeing. Obviously, you can argue that this could be a consequence of the endolysosomal problems they have, because that is stressing the cells, and stressing the cells will also have an effect on the mitochondria. So that was, in a nutshell, showing you how we use these IPS cell lines, how we're making these isogenic controls, and then studying the disease phenotype. And the work is mainly done by one of my PhD students, who is very talented and very dedicated. And we have done that together with Nanette, who's doing TEM. Then we also got a great hand from Benjamin, who is working at Bioneer and an expert in making these isogenic controls. So he helped us with the design. And we are all working at the University of Copenhagen with Professor Hüttel, who is an expert in electron microscopy. And then we're having various collaborations with pharmacology. We're doing these seahorse assessments. Then the patients have come from the Danish Dementia Research Center.
At Lundbeck, we're also doing functional studies of neurons, and in Pioneer, we're working on making these isogenic controls. Yeah, and it was funded. Obviously, we need some funding to do the work by the Marie Curie grant and Innovation Fund Denmark. Thank you.
Thank you very much for a very interesting talk. Does anyone have any questions? Yes?
Do you treat the cells with anything in order to mimic aging, as you talked about?
No. So we're basically just doing neural differentiation until week 4, so for 4 weeks. So they're even not completely mature neurons. But the advantage with these patients is that you can even see a phenotype in fibroblasts already, at least the enlarged endosome. So it's a lucky dementia, because a lot of these, if you're looking into Alzheimer's disease, oftentimes you don't see a phenotype because the cells, as you said, they're not matured enough. So that's a little bit more tricky.
Any more questions? Yes?
So you talked about you didn't have any, pure cell cultures. What do you do to accommodate this problem?
I wouldn't necessarily see that as a problem. I mean, we're getting about 90% glutamatergic neuron, and that really depends what you want to look at, because it's glutamatergic neurons, it's GABAergic neurons, which are most likely affected, and neurons also really need glia cells. So the field is kind of going a little bit backwards from making very specific artificial neurons, and most people now even try to do 3D differentiation and make some kind of miniature brains or organoids. So it really depends if your disease you're looking at is only affecting a specific, like for example, if you're looking in Parkinson's disease and you're only interested in dopaminergic neurons, then it would be a problem if you have not high enough enrichment. But even there, you cannot get completely pure dopaminergic neurons.
More questions? Otherwise, you will also stay later, so
Yes.
you can come and talk to her if you're shy now. You can think about it. Yes, so that brings us to our last speaker, coming all the way from Göteborg in Sweden, just to talk to you. He's from a company, Swedish company called Novahead. Yeah. You will be.
Yeah, thank you very much, Carola and the other organizers, for inviting me here. It's a great pleasure. As Carola said, I'm coming all the way from Gothenburg to here. It's great to be in Copenhagen, such a sunny and wonderful day. So you must be very tired now, and I hope you have fueled up with lemonade and chocolate and all that I saw on the left side here, because you have to stand my Swinglish now for almost half an hour. There we are. So even my presentation. So although I'm much older than the previous speakers, I have promised the organizers to tell you why I'm here, basically, my career, my life. So I studied biology in Gothenburg, a master's degree. Then, at that time, it was very easy to get a job in Gothenburg because AstraZeneca expanded almost as much as Novo Nordisk is expanding nowadays in Denmark. So you just knocked the door and you got a job. So I got a job in AstraZeneca and I started that. But after a couple of years, I realized that I would like to do something more in science, so I took a break and made my PhD in cellular biology and biochemistry at the Gothenburg University. And that took, as you know, 4 or 5 years, and then I was back in AstraZeneca again and worked there for a little while. But being in a big company, sorry Christian, was a little bit, I think, at least I thought that I needed to have a look at a small company. So I quit my job at AstraZeneca and I moved to a very small company. It was a startup, and I worked with this startup for 10 years, a little bit more, almost 12 years, and we sold the company to a French corporation, so it was a great journey to be in a small company, grow it. We were 60 employees at the end, and we sold it, so it was a nice story. But then I got a new possibility to start up a company, and that was 2 years ago, and that was NovaHep. So I'm going to talk to you about that today, and a technology that I hope you will find as interesting as I am doing. So let's skip that. So let's start with this very boring slide. You see some very fancy words here. Advanced regenerative medicine, individualized organs for transplantation, etc., etc. You see also the famous stem cells at the bottom end. So all these buzzwords and fantastic sentences, what are they all about? They are all about NovaHep's technology and business concept. So I would like to try at least to get you to understand what we are doing, and let's see how that will work. So first of all, of course, the acknowledgement that used to come at the end, coming now. So the technology is based on work from Professor Suchitra Holgersson in Gothenburg. She's an Indian lady, fantastic scientist. So it relies on tissue engineering and stem cells, as you will see further on here. What we are doing, we are really engineering organs, and we are personalizing organs. And that's very important. Why are we personalizing organs, and how are we doing that? I will try to explain, but simple just here is to avoid immune reactions. So let's see. This is, as you may guess, a heart. I think all of you know what is a heart, how that looks. If I ask you a question, what if I take away all cells from a heart? A heart without cells. What do you think will be left then? Well, not much, you think, a little slippery piece of tissue on the table. In fact, if you take away all the cells, you end up with almost the same heart. It's not the same because there is no cells around, but the shape, the three-dimensional structure is basically the same. And that is very important to try to get you to understand, because that is all about our technology. So taking away cells, you have the same, basically, heart. So the message here is that the cells in an organ, they are not responsible for the three-dimensional context of an organ. They are not. It's more or less exactly as a house. If you take a brick house and take off all the bricks from the house, it's not like a pile on the ground. It's still sort of a frame, a matrix, we call it in biology. So this matrix is very good to have because you can add new cells to this. Like the house, you can put up new bricks on it, different color perhaps. What we do, I will tell you in a second, is to add new cells. So the two very difficult words here, decellularization and recellularization, taking away cells and adding cells. Why are we doing this again? It is all about to have the organ go from an allogenic status, being a foreign status, to your own, what we call autologous status. And that's very important because then we rebuild a foreign organ to match yourself. And again, then we avoid the immune system. So this is what all is about. Just one slide to tell you that we have basically in our early research tested this on virtually all organs, so heart, liver, kidneys, pancreas, skin, nerves, etc., etc. And it's basically working the same. You can take off the cells and you can add new cells. And if you add the cells of the patient owns stem cells, you will end up with your own organ. So we design the organ for the patient. So the technology is great, I believe, but is it possible to go from a great technology into a company and a business concept, a company in a clinical phase? And how to choose here in the jungle of different opportunities? Should we start with the heart, with the liver, with the kidney, pancreas, whatever? Of course, we need a business model to do that. So I won't go into very much of details here, but we have decided to start our journey with blood vessels. There are several reasons for that. Blood vessels are among the most simple organs, you can imagine, compared to a heart or a liver, a blood vessel is more like a little tube. So it's a good start. But there are other reasons. I hope I will try to convince you on that. This is a blood vessel. So let's try to illustrate this again. I will really hammer into your heads now what we are doing here. So we are basically starting to rinse the blood vessel, because inside blood vessels we normally have a lot of blood remnants, etc. Then we add detergents, you know, washing powder basically. And the idea with them is they are lysing cells. We are killing cells. We are also using enzymes to take care of what is left after the detergents. So taking away DNA and remaining cells. I'm not sure you can see it, but we have basically this empty tube left again. Then we start the second process, being the recellularization. That is to illustrate, you have this, can I use this perhaps? Yeah, doesn't matter. This empty tube, and we introduce blood into this. So what I told you up to now is basically not magic, but this is a little bit of magic. To only take 25 milliliters of blood, you may know that when you are a blood donor, you leave half a liter blood. But 25 milliliters of blood is enough to recellularize a blood vessel. Basically, again, we are talking about stem cells. So stem cells are binding, migrating, dividing, and differentiating in this structure. And magic, in 1 week, we have a new blood vessel, more or less the same as we had before. So pointing out again that the blood is the trick, because there are other out in the world that are doing this, DNA recellularization. They're using stem cells, but they normally are taking out bone marrow or other good sources for stem cells, and they expand cells for weeks and months. And that is time-consuming, costly, and a bit tricky when you are facing the authorities at the end. But we do it with only 25 milliliters of blood and some tricks in 1 week. Yeah. This is again to summarize. We have the red guy, he's the donor, the foreign guy. He leaves a blood vessel, we decellularize, we recellularize with blood, we have a new blood vessel, and we can then transplant this blood vessel back to the patient, or to the patient, not back, to the patient. So, any reality, or what are we talking about here? Luckily enough, yes, because 3 to 4 years ago, we had some persons in Gothenburg that believed in the technology. We have a great guy, Professor Michael Oloison. He was very brave. He had patients. The patients were kids from 10 years down to 2 years of age. They had something called portal vein obstruction. Portal vein is one of the biggest blood vessels in the body, leading blood from the intestines into the liver. And if that vessel is failing, the liver is damaged, the liver is failing, and normally you try to do a liver transplantation. But do a liver transplantation on a 10-year-old or even worse, a 2-year-old kid is really a challenge, because you have to match the size of the organ, and the child will live without immune system for the rest of the life, basically, because you have to eradicate the immune system in order to do that. And that is the big challenge here. We are very reluctant in doing that with children. So instead, this guy, together with the founder of our company, he decided to transplant a new portal vein that was individualized with the NovaHeps technology to these 3 kids. And as you may see here, that was a great success. Follow up, 3 to 4 years, these kids are still carrying the new blood vessels. The liver started to more or less immediately to recover, and everything was good. The big scientific height here, or news here, that was we did not use any immunosuppressive agents at all. So that was sort of a proof of concept here. Next chapter, because portal vein obstruction is a very relatively rare disease, so not so many patients. We are a company. Our entire bodies are full of blood vessels. So we looked into this and thought, which one is best to start with? And there is a rather obvious alternative, according to us. And that is to start with blood vessels with valves. So in some of our blood vessels, especially the big veins, the deep veins inside our legs, they are carrying valves that are opening and closing and allowing or not allowing the blood to pass. If those valves are damaged, you are affected by something called chronic venous insufficiency. You will see that in this slide. Quite nasty. You will have pain in your legs, you will have swelling, but ultimately you will end up with quite nasty leg ulcers like this. And the reality for this patient is that there is no treatment today because there are blood vessels on the market today. Most of them are artificial blood vessels. You may have heard that one can design blood vessels in a lot of different fancy material. One good example is the American company Gore. They are making blood vessels out of Gore-Tex. So they are great in one way, but there are two big disadvantages here. They are not having a biological surface, so they are affected. These artificial grafts are affected by thrombosis a lot, so they have to operate the patients again, exchange these blood vessels, etc. And importantly, no one, to our knowledge, has solved an artificial material for a blood vessel with functional valves. For us, it's easy because we just take a donated blood vessel that are already carrying valves, and we and recellularize, and out we have a new blood vessel designed for the patient. So we believe we have a rather unique concept. We are replacing these veins with valves, treating chronic venous insufficiency. Interestingly, we have 2.2 million patients only in the Western countries. These patients are very often on sick leave. They are already retired early, as we say, at least in Sweden. And altogether, they are consisting of an enormous socioeconomic burden. So it's a virgin market for us of about 2 billion Swedish crowns, with today no competition. We haven't heard about any competition, at least. So our strategy then, the immediate goal is to industrialize this technology. To do it in a research lab can be done, but we believe we should build almost factories that are producing blood vessels, so thousands of new blood vessels. Then we really have to industrialize this, get it extremely robust. So scale up, robust, simple, cheap, and fully clinical compliant. This is an enormous challenge for us, but it's also fantastically fun. We are now planning to start a clinical trial, a real clinical trial in Oslo early next year for these chronic venous insufficiency patients. We hope that will be some really exciting study that can be followed by others. So we believe this will become a completely new industry. I have now focused most time of my talk here on blood vessels, but I would, before stepping down, give you some other examples here. So this is a kidney, and what you see on the left side, of course, is a normal kidney, and here you can see the decellularized kidney. It sounds easy when I explain what we do, but remember that what is important here, I would refocus on that again, that we do the decellularization in a very gentle way. It's rather easy to kill all the cells. That can any of you do at home, I think, with some washing powder and other things. But then you will also destroy the matrix. You will also kill something that is extremely important. And in this, two things that are extremely important. You will destroy the substructures. Here you see the glomeruli in the kidney, and you will immediately destroy them. It took us about 2 years to go from start until we understood how to decellularize without destroying the substructures. But what you also take away or destroy is all the factors that are bound to this matrix. So it's important to know when we take off the cells, we have the matrix, that is basically extracellular matrix proteins, but bound to this matrix is also all the factors that are steering the stem cells. If you remember the two previous talks, we need factors that are steering stem cells. And I have actually, I didn't say that, but worked together with Novo Nordisk in a diabetes stem cell project for many years. more years than Christian indeed. But they are doing a fantastic job in Novo Nordisk, but it is a challenge to find all the steering factors. What we are doing nothing. We only use what's already there. So we keep all the factors and we let them steer where they are in the perfect three-dimensional structure, the cells. So when we add the stem cells, everything is served, sort of. This is, of course, a must in this country to talk about pancreas because we are in the homeland of Novo Nordisk. So this is a pancreas with cells and without cells. Again, more or less the same. You have the scaffold, but you have no cells. So this is a very early project for us. We hope that we can work together with Novo Nordisk and Danes in the future in this area as well, but really early days. Just have to mention before I leave that one can also use our technology for in vitro application. One of the big challenges for the traditional drug industry, that is to early in the development process identify negative effects of the compounds one is developing. And the most important organ is the liver. So what we do is we take a liver, the native organ, we decellularize. And I hope you can see how beautiful this decellularized liver is with all the vasculature still perfectly intact. Then we can add stem cells, and to make the two previous speakers happy, we are using a lot of iPS cells here because we believe to have a perfect in vitro model in the future, iPS is the answer because you can design the disease model, you can design the genetic background, and as soon as you know how to do, you can change your model as you wish. So we recellularize with stem cells, and then we can either use a whole liver or we can chop it up in small pieces and do these type of assays that is very common in the industry. And test drugs for interactions or metabolism or other effects in human liver system. A good human liver in vitro testing system is not available today, so we think there is a great need and interest for this. So you are all young students, so I think it's fair to say that the future is a great challenge. To build up a company on a sexy and fancy technology may be very fun and interesting, but it is a challenge. You need to find money to do that. You need to have committed personnel. I think we are on our way to start that journey in Gothenburg. And you need to collaborate. You should not sit down in your office or in your lab in Gothenburg and believe you can rule the entire world. You need to come out to DTU, for example, and collaborate. So now in Gothenburg, we have recently got new owners into the company. They have financed us even better. So now the journey is on and we are building the company. So I would like to, by that, say that, or thank you so much for your attention. Tell you that you can read more, of course, at our homepage, or you can ask me questions now. Thank you very much.
Thank you very much. This was very interesting. So if anyone has questions, feel free to ask them. Yes?
You talked about upscaling your production of the blood vessels. Like, I imagine a factory like cadavers in one way, blood vessels out the other way. Do you have enough donors?
Yes, very good question. And I almost felt that you thought this guy is hiding something, but I'm actually not. But right now we are using human, it's a terrible word, isn't it? Cadaveric donors. So we are basically picking up organs from dead donors. They are, in some cases, donated to research or to guys like us. So we are taking that type of material. And we believe that for quite some long time, we have enough material. You can imagine that a dead person can donate a lot of blood vessels. And we only need a little piece of a blood vessel to do what we are doing. So we believe so, yes. However, it's a good question because it's a little bit of a tricky procedure to go to the hospital and pick up this material. So in parallel, we're also working on what we call then xenogeneic material. So we believe that the scaffold, well, can be from a pig or a cow in the future. So we can use a pig vessel, decellularize it, and recellularize with human cells. In the same way that the recipient here do not identify that this blood vessel from the early beginning was from a foreign donor, we believe that the recipient do not either will identify that it's coming from a pig. We have very positive findings from the labs. So in a couple of years, I believe we are switching from human donors to animal donors, basically.
Yes, more questions?
Ladies first. That was not a lady.
No. Totally sorry. Since your company uses stem cells, which has qualities such as unlimited cell proliferation and high telomerase activity, isn't there a higher risk of developing cancer since these are like pro-oncogenes?
A good question. I think again, I don't think I have all the answer to that because we haven't used a lot of different sources when it comes to our blood vessel program. And the reason is that we have been so efficient in using human blood. And I mean, we are using not only human blood, we are using the patient's own blood. Exactly the cells that are anyway circulating through the veins and arteries of that patient. So we haven't seen any signs of that when using whole blood and the stem cells in whole blood. But it may be when we change. And if we would go into iPS cells for clinical application for the future, that is, of course, something we have to look very carefully into. But no signs, but we haven't studied a lot.
Yes?
Okay, so when you move on and want to grow hearts and livers and whatever, do you still think you're going to be able to seed the scaffolds with blood, or do you think you have to go in and use induced pluripotent stem cells?
You have so many good questions. Again, I don't think we have all the answers. I don't think we can use blood for all different organs, no. I think we can use them for some organs. We have early data for a couple of other organs. We're using whole blood, but we are also using other sources of stem cells, indeed, when recellularizing other organs. So the question is, I don't know fully, but my guess is that we need other sources also in the future.
Thank you. So, do you do research in artificial scaffolds or only in natural scaffolds?
We only do it in natural scaffolds. Okay. We believe that is our competitive edge to do it. And to repeat myself, the artificial scaffolds, they are really our competitors as of now. We have, as you may understand, we have an IP portfolio in the company that we are using, and that is only sort of covering biological material and biological scaffolds. And also, there are already artificial material out there for different purposes. In general, all these materials still have problems because they are not fully biocompatible. We believe again that we are not, it may look like rocket science here, but we are basically only using the Mother Nature's own structures here. So taking off and adding new cells in an easy way. So answer is no.
More questions?
Thank you. You said you had an IP portfolio, but how do you protect this?
Very good question again. So we have indeed an IP portfolio, and patents are covering key steps here. So for example, we believe, or we partly know, that the use of whole blood for recellularization of these type of structures, that is our property. So we have very strong patents worldwide that we believe are far ahead from everyone else. But then we also, as a company, I've learned working with stem cells many years that in the labs when working with stem cells, you have a lot of tricks and green fingers and things like that. We used to, or I should rather start to say that there is a big disadvantage with patents. Namely that after 18 months after filing, they will be public. And everyone can read exactly what we are doing. And there are countries that are not following patent laws. There are other reasons for keeping part of the process as a trade secret. So that is what we are doing. We have a combination of patents and trade secrets. And trade secrets are the tricks I refer to. We never publish them, we never patent them, we never show them, as you may imagine. So they are our own things. So we have to rely on our employees, etc., and have good agreements with the employees.
Do you see the hype about 3D printing in biological matter as an opportunity in the future for making the matrix?
Yes, indeed. That is something we are doing as well, because bioprinting is still quite a way in the future, I believe. So, but in 20 years or 15 years, we may be very good in bioprinting. You know, bioprinting is that you take biological material, cells or matrix proteins, and you print them. The problem right now is that even though these tubes, when it comes to stem cells, or the heart scaffold looks rather simple, they are extremely complicated. They are organized in an extremely complicated three-dimensional structures with all proteins or other components in a perfect order. And it will take a long time to understand. But for simple organs like blood vessels, I'm sure that it will be an opportunity for having the scaffolds in the future, and perhaps even also the cells on top of it. Definitely. So bioprinting we are following and partly work with, but still on a early stage. Sort of.
So production-wise, are you looking into automated production, or is it still with labs and lab benches and so forth?
Very good and very evil question. No, production and scaling up is really a challenge, because we need to lower cost of goods. We really need to lower the amount of personnel needing for every step here. So I think it will be really a stepwise process. We are going from today an extremely manual process, really personnel with green fingers that are doing this. But we see that we can industrialize many steps. And again, that is something we are learning from the field of stem cells that has evolved tremendously with robots, with the bioprinters, with a lot of other stuff. So I think in more and more, and in the future, I think we can even have robots handling part of this process. But again, quite a bit away in the future still.
More questions? Yes?
You are exercising today, yeah?
Yes, you're deciding to...
Yeah, I was just considering about the clinical trials. How will you ensure with using a matrix from a donor that you won't transfer diseases to the patients you're trying to treat?
Thanks for that question. Of course, starting a clinical trial is nothing you do overnight or in a few weeks. It's a long, long process. So exactly as you say, you have to control your process. You have to control the whole procedure from the donation in the hospital of the dead, diseased material into transfer to us, and it should be controlled all the way that we do not carry diseases from the donor, etc., all the way from HIV to hepatitis and all the other stuff. And also artifacts coming from the surrounding in our labs. So we have to protect ourselves from the material and the material from us, and we have to control it. So it really, I mean, piles of documents, and we have a quality system that we have more or less just started to build up. But in order to get a final go for the clinical trial, the process has to be extremely controlled. So we believe we are on the way. We have a lab and personnel that is doing that right now. And as I said, early next year, we are starting a first clinical trial. That is a small trial. We are estimating 12 to 15 patients. So it's a classical early study. But we believe that if we control the process, it's a relatively safe study to take off a blood vessel and add a new one. And surgical-wise, it's a really simple surgery to do that.
more questions? Otherwise, you will stay a little longer, I hope, for questions. Yeah, so thank you very much, and to all the three speakers. Thank you very much. You get a... and sorry, I forgot the... So yes, thank you all for coming. It's really been wonderful to seeing so many people here. We never had so many people for a lecture session before, so it's really exciting. And remember, find SBE on Facebook and talk to the speakers if you like, or any of us if you have anything to talk about. So yeah, thank you and have a good night.